Welcome, everyone. Thank you for joining us for today's webinar, Precision Medicine for Mice, Digital Biomarkers Transforming Drug Discovery. It is presented by Laura Schaevitz, PhD, the Chief Scientific Officer at Vium. My name is Judy O'Rourke, and I will be your moderator for today's event. We are delighted to bring you this educational web seminar presented by LabRoots. LabRoots is the leading scientific social networking website and producer of educational virtual events and webinars. Before we begin, I would like to remind everyone that this event is interactive. We encourage you to participate by submitting as many questions as you want during the presentation. Simply click on the Ask a Question box located on the far left of your screen and type your questions into the drop-down box. Our speaker will respond to your questions via email following the presentation. If you have trouble seeing or hearing the presentation, please click on the Ask a Question box to let us know that you're experiencing a problem. This presentation is educational and thus offers continuing education credits. Please click on the Continuing Education Credits tab located in the top right corner of the presentation window and follow the process to obtain your credits. Now, without further ado, please join me in welcoming Dr. Schaevitz. I will now turn the presentation over to her.
Thank you, Judy, for that fantastic introduction. Today, I'll be talking about precision medicine for mice and how we at Vium are thinking about digital biomarkers as a way to transform how we do drug discovery. During this presentation, I'll be covering a number of different topics, and just to give you a preview of what that looks like, we'll be defining digital or biomarkers, and then what's the difference between a biomarker and a digital biomarker. In addition, I'll be going through some use cases where we'll look at how high-frequency data captured through digital biomarkers provides more objective, sensitive, and meaningful readouts of disease that better translate to clinic. And in that section, I'll actually be showing some data from clinical studies, as well as some of the work that we've been doing in the animal models to show how some of that can actually translate directly. And then finally, I'll spend a little bit of time on how we're thinking about the future challenges and opportunities, especially around digital biomarkers and preclinical disease, and how we can potentially continue to improve preclinical studies and their translatability into the clinic. So I wanted to start with what are biomarkers? And this definition comes directly from the biomarkers definition working group from the NIH. Here, biomarkers are classified as a characteristic that is objectively measured and evaluated as an indicator of normal biological process, pathogenic process, or pharmacologic responses to a therapeutic intervention. And why are they so important? And to talk a bit about that, I wanted to bring up one of our standard views of how drug development happens. So here you see a very standard timeline in which you start very early on with thousands of compounds in an early drug discovery phase. Those lead candidates will then get taken into the preclinical phase. So you'll have a much smaller number of compounds in the preclinical. And then only the best from preclinical will actually be transferred into clinical trials. And at the very end, when you've started with tens of thousands of compounds, you end up with potentially just one compound that goes through FDA review and approval. Biomarkers play a really important role in this drug development process because, in essence, think again, biomarkers are ways of tracking whether or not a drug is having a positive effect on a particular area or mechanism of action. And so what many have done are actually looked at particular ways of measuring whether cells are reacting. And those same cells could be the cells that are within animals in the preclinical phase. And those same responses can be also monitored in clinical trials. So this is a way of being able to marry from basically the beginning of drug discovery high throughput screening all the way through clinical trials, whether or not a drug is affecting a target in a way that it is expected. So in essence, saying that again, this now provides you a measurable way to look at the effects of your investigational drug throughout the whole development process. So you know if it works all the way back in the drug discovery section or area phase, that it should work in preclinical and then of course in clinical trials. Biomarkers are really important, and there are a number of consortiums that have been created, including a joint FDA-NIH Leadership Council, in order to identify what's required for a biomarker, what those endpoints should look like, and how to quantify them and validate that they are effective all the way from, again, early drug discovery through the clinical trials. Generally, biomarkers are thought of as some physiologic characteristics that can be monitored, but mainly they're molecular in action. For example, looking at specific molecular markers all the way through the cells into the humans. But there's this new class which is starting to emerge, which we call digital biomarkers. And I throw up here a definition which is not an accepted or standard definition, because to date there is no unified definition of what a digital biomarker is. But what you'll see here, digital biomarkers are physiologic and behavioral measures collected now through connected digital tools that are used to evaluate those same processes. So we're talking about biological processes, pathogenic processes, pharmacologic responses to a therapeutic intervention, for example. Now, the key here is through connected digital tools. And digital biomarkers are generally thought of primarily in the clinical space. And actually on this next slide, I can show you there's a huge ecosystem of wearable and implanted sensors which are digitally connected. And the way we think about digital connection is actually through your smartphones or your computers. And so these are sensors or wearables that are on your body that transmit all of that information continuously or at high frequency to your smartphone up into the cloud, where it can be looked at more broadly. So this is just an example from a Nature Biotechnology paper published in 2015 of all of the different digital biomarkers that are out there and currently under development. So you can see things like being able to monitor ECGs, heart rate, respiratory, glucose, for example, or even compliance of taking pills. So there's a sensor inside of pills that will tell you whether or not a pill has been ingested at the appropriate time. So we have this whole ecosystem of digital biomarkers that have started to be developed. The question really is, how do we use that to better understand disease and how we can potentially, in a precise way, affect individuals? So on this slide, I've showed just a few examples of papers and where these digital biomarkers are coming from. So you'll see in the top left-hand corner, here we're looking at, can you identify stage of multiple sclerosis using just physical activity monitoring? Or can you look at chronic obstructive pulmonary disease by listening to the sound of a person, how they sound when they're actually breathing? Can you look at heart using ECGs that in this example, you actually put your fingers on two little pads and it measures the ECG. Now, of course, the Apple Watch will do that for you as well. And then there are also some work done in Parkinson's disease where simply you tap the iPad. This is just a few examples. in many, many different therapeutic areas, sensors are now being used to see if we can learn more about the disease, the progress, and identify better ways of actually diagnosing and, again, seeing whether or not there's a therapeutic response when given a drug. Turns out that many of these digital biomarkers exist in the preclinical space today as well. We just haven't previously thought of them as digital biomarkers. So what are some of those examples? We can monitor physiology in rodents as well as in dogs through implantable sensors, telemetry systems. So what I'm showing you here is a DSI system, although there are many different systems out there that do that, where we can measure ECG or EEG, body temperature, glucose readings. Again, these are implantable sensors, and you can get this information from the animal pretty much continuously over a defined period of time. We can also look at metabolic parameters in these animals. So here's just one example. Again, there are many companies with these systems. Here we can measure oxygen consumption, carbon dioxide production, respiratory quotient, food and water intake, information on wheel running, activity, body mass, sleep time. So you can see how these are directly correlated to what we would potentially be interested in measuring in the clinic. We can also monitor specific behaviors. So here's a Clever-Sys system, an IVC rack from them, where we can measure a number of different specialized behaviors such as drinking or foraging and the percentage of time that they do each of these. And then the last system is actually a system that I've been directly involved in developing. And this is a smart caging system where we can monitor both behavior and physiology. And so here we can monitor motion, breathing rate, wheel running, circadian rhythms associated with that wheel running as well as starting to work on being able to, in real time, read out information on those animals. So when we think about these two types of systems, the data that you're getting from the humans, from their smartphones and their watches, their Fitbits, as well as the data that we've been collecting for many, many years in animals and in home cage monitoring systems, such as those that I showed you here, or telemetry, where we're looking at ECG. The idea here is that we can actually start to better bridge the gap between preclinical research and clinical research by actually looking at the same behavioral or physiologic outcomes between the two. And so if you focus on the right, here is here's a digital twin concept. So the idea is we're taking all of these biomarkers from the human. Here we show respiratory rate, cardiovascular activity, neurologic, metabolic. We take those readings from the mouse in exactly the same way using exactly the same types of technologies. We have diseased models for the mouse, diseased models for the human. We can build potentially better models and be able to track better from early on whether a mouse is having a positive response to a drug that would hopefully help us better predict, again, whether a human would have a positive response from a drug. So today I want to focus on two case studies. The first is in multiple sclerosis. And here in humans, we've talked about, or there are papers out there on how activity levels or step counts can help predict disease severity. And I'll show you some data that we've started to gather thinking about the same problem in mouse models of MS. And then the second is in a rheumatoid arthritis model, or in rheumatoid arthritis. And again, in the human, there have been a number of studies looking at how duration of 5-time sit-to-stand tests actually predicts stiffness and pain in individuals with rheumatoid arthritis. And we're going to look at, in the rat model of rheumatoid arthritis, are there similar measures that we can assess that would help better develop or build better models that we can then test our compounds against. So starting in multiple sclerosis, let me tell you about a study that was published in the Journal of Digital Biomarkers just this last year. The study was a fairly simple one. The objectives were to advance the literature on use of free living physical activity tracking from MS patients. So these MS patients wore the Fitbit One wireless activity and sleep tracker, which you see over on the right side of your screen. There were 114 self-selected patients. They uploaded their data from the Fitbit One, as well as they self-reported on their multiple sclerosis rating scale, which included a number of different features such as ability to walk, upper limb function, vision, speech, swallowing, thinking, memory, cognition, sensation, burning, and pain. Here we'll be focusing primarily on the results from walking, but also more generally from the multiple sclerosis rating scale. To just go right to the end, or what they found, what you can see here is that step count well predicts disease severity in this particular example in MS patients. So I've just pulled from a single figure from this paper, but here age of the individuals is not different, gender is not different, disease duration is no different in terms of overall step counts. So here you can see age, and then those numbers in the column, the 3,000s, 4,000s, 5,000s are the number of steps that that individual or that group had taken on average. Once you get down to the MSRS, this would be the multiple sclerosis rating scale. You can actually see that there are statistically significant differences as you move from the individuals that were reported the least disability up to those individuals who reported the highest disability. So specifically here, you can see that those with the least disability walk on average around almost 6,000 steps, whereas those who are in the highest disability quartile are walking on average around 3,000 steps. So almost half as much. And then if you further break that down into specifically how they rate their walk score, that actually pulls apart even more. And you can see those that report no disability around the ability to walk report taking almost 6,700 steps, versus those who have moderate disability are taking only around 2,700 steps. So here, it's quite clear that just the number of steps that these individuals take fairly well indicates how disabled they are, or they report being. What's even more interesting is that the predictive power increases even more when the authors of this paper actually average step count over multiple days. So this is a really important piece, and we'll talk about it as we get into the animal data as well. But many of us have days of higher activity and days of lower activity. And so the idea here is that if you actually average the step count across multiple days, 2 days, 4 days, or 7 days, you'll get a better sense of an individual and their overall activity levels. But that means that you can't gain this type of data unless you're actually collecting it continuously, and you gather lots and lots of data on the same individual over time. And this is where the digital biomarkers really show how important they are. This is not information that you can get from a patient who visits a clinic on one day, as an example. So now if we move on to the animal models, let's look at how these digital biomarkers, i.e. step count in the human, can potentially translate to what we're seeing in the animal model. So here I'll show you data that we've collected in our own lab on the Cuprizone mouse model. Cuprizone is a model in which we feed the animals Cuprizone, which is actually a metal. It causes rapid demyelination, and then followed by remyelination as you actually remove the animals from the Cuprizone. It's a model that's commonly used to assess kind of remyelination or to study remyelination. It recapitulates many of the characteristics of MS pathology, but it does not, and one key critical difference here is it does not have an autoimmune component. So what I've just put up on the screen is a timeline just to give you a sense for how the experiment was run and what was measured at any given time. So here we have an acclimation period. So we always want to bring animals into the facility and get them used to the facility, because what we find is that they, in the first few days, their behavior is not normal. So again, it's just as we were talking about with the humans, give them a little bit of time to get used to their surrounding. Then we put all the animals on either a Cuprizone chow diet or a control diet, and we monitor them for 6 weeks. And then at the end, you'll see data where we've actually removed them and put all animals back on a control chow. Now, in a model like this, you would typically measure one of a couple of things. You might measure body weight. You would likely measure a behavioral assay. So much in the same way as we've just talked about step count being important in patients with MS, you would expect that you should be able to see motor deficits in these animals, and that might be done on either a rotor rod or in an open field. And then you might also do histology. For the purposes of today, I'll just focus on the behavioral assay. And the one that I'll talk about here is an open field assay, in which you actually take an animal and put it into a big box, which you can see on the right, or sorry, on the left of the screen. And the animal will run around the box for somewhere between usually 15 minutes all the way up to an hour. You'll often measure the distance that the animal travels and then the speed at which they travel around that box. It would be done infrequently, so you might do it once over the course of a study. You might do it a couple of times. It's also usually run during the daytime, when an animal, or a mouse in particular, would not often normally be awake. So you'll wake them up and then put them in this box, and it's a novel environment. So you can also imagine there might be some differential effects by the fact that the animals are exploring this apparatus as well. So when we look across studies, here's just a smattering. I took a quick look across in the literature, and you can see, and I've shown you here, on the left, we have studies where comparing normal animals to animals that are fed Cuprizone, they show significantly reduced activity in the form of less distance traveled around the open field. In the middle, there are many papers in which there's no change in the activity levels in these animals, so whether it be by speed or by the path length. Conversely, there are also a few studies out there in which, when you compare the control animals, which it's a bit hard to see, but those are the ones in the darker black lines, to those that received Cuprizone chow, those of course are the lighter gray lines there, they're actually, those animals on Cuprizone are more hyperactive than animals on normal chow. So, when we talk about reproducibility in animal research and whether or not you see a positive response to a compound, when the literature shows such varied responses, it's really quite challenging to know which is true. And if you see a positive effect of your drug or you don't see a positive effect, how to interpret that data. So what we did in our lab is actually put the same model into this home cage system. So here we bring animals into the facility and they go directly into the system. We are monitoring continuously, again, the motion, breathing rate, and overall activity levels on these animals continuously throughout the whole entire, 9 weeks of the study. It's not really a novel environment except for in that first day, because after that it becomes a fairly standard environment for them, except for in the case of cage changes. We can run them during the daytime or the nighttime. We can look at data continuously. Here again, we're looking across the whole span of the study. And rather than looking at just 12 hours duration, now we're looking at the full 9 weeks. So what I'm showing you here is data that we collected specifically just after cage changes, and we changed the cages every 14 days. So to orient you to this, the dark gray lines are those animals on control chow, and the light blue are those on the Cuprizone chow. We're looking at the average daytime motion. So again, trying to compare it to what has been done in the open field. Where you see the dotted lines, that first dotted line is where we transition from control chow on all animals to the Cuprizone or the control. And then that second dotted line is when we transition all the animals back off of those that have been on the Cuprizone chow off of the Cuprizone chow. And what I hope you can see is in the very beginning, when we look just at the response after the cage change only, there's no real difference between the groups. Then when you get out 14 days and after being on Cuprizone chow, we actually start to see some suggestion of hyperactivity in those animals on Cuprizone chow. So this matches that last data set that I had just showed you. And then when we take the animals back off of the Cuprizone chow, again, now they appear to be somewhat similar to the control chow animals. But if we take into account all of the daytime data that we've collected on these animals, so here now you can see all of the data averaged during the daytime, that pattern of hyperactivity seems to go away. So if you compare here now, the majority of the days between cage changes, there's maybe some evidence of hyperactivity, but for the most part, the animals move around very little in comparison to those cage change days. And it seems to be very similar between those animals that were on the control chow and on the Cuprizone chow. So now potentially we've recapitulated what you saw on that, those first open field charts once the animals had been potentially habituated to the open field. But even when we're looking only at that daytime motion, we're still only measuring a portion of what's actually observable in those animals. So remember when we go back to the paper where we were looking at the number of steps in the MS patients, we weren't looking at the number of steps at night when they should normally be asleep. We were looking at the number of steps that they might take during the day. So the question that we then looked at in terms of this data is, are there actually differences in motion levels at night when animals are naturally active? As we would hypothesize that that would be the data that would be most applicable to what you were seeing in the patients with MS. And so here I'm showing you now, this is averaged across the course of that those 9 weeks, the nighttime motion for, again, those animals receiving control chow are the dark lines, and those animals receiving the Cuprizone chow are the blue lines. So prior to the animals receiving Cuprizone chow, we see very similar activity levels between groups. You can see prior to the Cuprizone chow being given, that increase in nighttime motion over time, and that actually is associated with them habituating to and getting used to the cage. But then once we transfer a portion of those animals onto Cuprizone chow, there's a significant decrease in the overall activity levels starting even 5 days after we start moving the animals onto the Cuprizone chow. What's interesting, and you can see that that goes all the way up until the 14 days. What's interesting is right around those days where we do the cage changes, again, we're inducing novelty to those animals. We see a return to normal even in nighttime activity or motion levels. And then as they get used to the cage, or at least what we think is as they get used to the cage, their activity levels drop again, and so on and so forth. So as we think about it, we really think that this decrease in the nighttime motion that we see not around those cage changes is probably most directly translatable or relevant to what you would see in the human patients with MS. So where's the value then of the digital biomarkers? So some of the areas that I talked about here were that traditional measures really only observe a small portion of what's actually observable. So here, what's most relevant is the data at night after the animals had gotten used to their environment. The more data we collect allows us to better characterize the animal. So if you had followed as we went through looking at various aspects of the motion data in the Cuprizone model, we actually nicely recapitulated all of the data that had been found in the literature in the open field. It would be interesting to go back and to determine whether or not you could actually recapitulate those results in each of the open fields. But it does suggest that the more data we get, the easier it will be for us to accurately characterize the animal and know what pieces should directly be compared when you're thinking about therapeutics, for example. And then last but not least, and I think this really ties from the previous bullet point, which is digital biomarkers, which are acquired under natural conditions, are going to more closely resemble the predicted responses. So when we're not physically handling the animals or interacting with the animals, we're more likely to see kind of the natural state of the animal, which in a very sensitive model like the Cuprizone model, where it's not always easy to get a reproducible response, it's these times where we would expect to see better or more easily identifiable responses that we can assess therapeutics against. So the second model that I want to talk about is predicting pain and stiffness in rheumatoid arthritis. So again, just quickly to go through some published data in humans with rheumatoid arthritis or patients with rheumatoid arthritis. I want to show you just on the next slide a single chart in which they're looking at the importance of being able to monitor the duration of 5-time sit-to-stand tests. In this case, the study, the objective of this study was to demonstrate that the measurements collected from this 5-time sit-to-stand test would enable them to derive an objective signal related to morning pain and stiffness that could be monitored without actually having to physically ask the individuals to report out on their pain and stiffness. And more importantly, they note that in the clinic, it's very challenging to actually be able to assess morning pain and stiffness, because by the time the patients actually get into the clinic, that kind of first morning pain has actually gone away. So the question is, can you use one of these digital biomarkers to be able to assess that in the home or kind of natural state of the patient? So here they describe data on 30 participants the participants were all given one of these actigraph link devices, which looks like a watch, but it actually contains an accelerometer, which can monitor the speed at which the individual is moving at any given time. So they record throughout the full 20 days that the patients have this device, as well as they ask them to do these 5-time sit-to-stand tests, a couple of, I believe they asked them to do it 3 times a week. So on Monday, Wednesdays, and Fridays. And that just involves them crossing the arms over their chest and then going from a seated to a standing position 5 times. They also asked them to report out pain and stiffness. So what they wanted to do was relate the reported pain and stiffness to the duration that it takes them to do these 5-time sit-to-stand tests, and hope that they can actually pull out biomarkers from this actigraph link device that would allow them to basically assess that without having to get all of this other data. So here's a very challenging graph to read, but in essence, what you're seeing here on the y-axis are those patient-reported scores. So the higher the score, the more pain and stiffness the patient reports. And then on the bottom is the normalized sit-to-stand test duration. So this is the amount of time that it takes them to do the test. And what you can see is that in general, the more pain that's read out, or pain and stiffness that's reported, you see a longer duration in actually being able to do this test. And so in this paper, the authors talk about the ability to actually distinguish morning and evening stiffness, and more importantly, that morning stiffness is more severe as patients report, both in the 5-time sit-to-stand test, as well as in what they can pull out of this actigraph monitor as well. So how does this relate to rheumatoid arthritis models in the animals? So in this case, what I'll show you here is data from a collagen-induced arthritis model. So here we're using a very standard model in which we give animals collagen to induce arthritis in the hind paws of the animals. It's one of the most commonly used models in rheumatoid arthritis research. It nicely recapitulates disease symptoms and other features of the disease. Much like in the Cuprizone model, we have an acclimation time period. Then we induce the animals through a number of these injections, and then we'll monitor them over time. And that is what I show here as the treatment phase. Traditionally, in a study like this, you would measure a number of different things. You would look at joint thickness. So I'll show you in just a second, but you take calipers and measure the ankle size of the animal. You might do a subjective scoring of arthritis, and you would take those paws and look at histology in the animals over time. So again, while each of these are important, none of them are directly applicable to the common kind of outcome measures that are looked at in the humans in the form of, reporting out on pain and stiffness and whatnot. So just looking quickly at arthritis scores and thickness measurements. So here are your standard assay parameters. You would ask a research associate to go in and to actually look at the paws of the animals. It's a bit of a small picture, but depending upon how many toes appear to be inflamed and whether the inflammation has actually gone up the foot, you would give the animal a different score. And then you would take that same animal and measure the joint thickness using a pair of calipers, and you can see what that looks like on the right. Challenge with both of these is that they're fairly subjective, and in terms of caliper measurements, one technician may squeeze a different amount versus another technician. So what I wanted to do is quickly just show you some data that we gathered from our own lab. So here you can see joint size measured across the course of this 17-day study. And I'm showing you two control groups in the green and the light blue. So you can see really no change in joint thickness across the full 17 days. And then I have two groups, an A group and a B group. Both of these received collagen, but my question to the audience here is, which one received the more effective drug? Give you in 1 second and then show you that actually both of them are vehicle animals. So what I really wanted to just point out here is that research associate number 1 took the joint thickness measurements from the first set of animals, and research associate number 2 took joint thickness measurements on a second set of animals in a separate study. So this just points out how challenging it is to actually reproduce across studies and potentially across technicians, trained exactly in the same way together on the same measurements in our lab. So what we wanted to do was create an algorithm to identify the disease without having to rely on these subjective measurements. So what I'm just showing you on this slide is a kind of broad strokes overview of how we actually did that. We took from more than, I believe, 150 different animals, those joint size measurements, as well as the arthritis scores and histology measurements. We took all of those together as our expected results for each individual animal. And then we took the corresponding motion data and we extracted a number of different features from it. For example, how active was the animal during the nighttime? How active were they during the day? How fast were they, as examples? And we looked to see which of those features best predicts the expected results. So in essence, what we discovered is that the maximum speed at night actually is the best predictor of some of the standard measures that you're looking at, histology being a really important one, arthritis score, and joint measurements. So in essence, what we're showing here is what we're saying is that the more the arthritis the animal has, the less quickly they can move around the cage at night, which, remembering back to those patients in the clinic, seems to be a very direct measure. Now we're talking about, in that case, we were talking about how quickly could they complete a sit to stand test. Here we're talking about how quickly they can move around the cage. So on this slide, I'm just showing you again the data. You'll notice the joint size and the arthritis score is identical from what you saw on a couple of slides ago. And overlaid on that now in blue are the digital arthritis index that we created using this automated algorithm. And I think what's most telling here is that when you look at joint size and the arthritis score, it would suggest that the first experiment had a more severe response than the second experiment. But it actually turns out that in reality that was reversed. So we see more severe histology in the second experiment than in the first experiment, and the digital arthritis index that we created recapitulates that. So once we created the index, we actually used these digital biomarkers to screen a number of compounds. And this just shows you the responses from 9 of those compounds that we screened. You can see that the animals that received vehicle have no real change above baseline, and that those blue lines show you improvement over the vehicle animals that had received the collagen, so those animals that had arthritis. And so animals that received MC105, for example, MC110, MC111, and MC114 all showed improvements. And all of this data can be found in our publication in Frontiers in Pharmacology. But what I really wanted to point out here is that if you look across all of the various measures, so here I'm showing you each of the compounds, their total histopathology score, their score on the digital arthritis index, inflammation, arthritis, and joint size. And we rank order them across those 9 compounds. You can see on average, all of these rank order animals in exactly the same way. So MC105 shows improvement across all of the various measures. The MC103 appears to be pretty good across most of them. You know, there's some slight differences as you rank order them across each measure. But there are 2 compounds that really stand out as different. And I just want to take 2 minutes to highlight these. The first is MC114. And what's interesting about this compound is that you see improvements. So statistically, they were better than the vehicle when you think about inflammation, arthritis, and joint size. But then when you look at total histopathology score in the digital arthritis index, that would suggest that there was no statistically significant improvement. So here, this digital measure of monitoring arthritis is better at predicting histopathology than some of those standard measures in life in the animal. The last one is MC111, which is actually quite interesting because there's no real improvement across any of the measures, histopathology, inflammation, arthritis, or joint size, but we actually see an improvement in the digital arthritis index. And what's interesting about this compound is it's actually a corticosteroid. So this one might actually make sense as well. In this case, we're probably relieving the pain. And so we're seeing an improvement that appears to be functional, even though it's not pathologically improving. So what's valuable in this case about being able to use something like a digital biomarker is you can see not only changes in kind of pathology and overall changes in joint in this case, but you can actually see a functional improvement. So taken together, we get far more better, interesting data when we take digital biomarkers and we compare them to standard biomarkers. So just quickly to go through this, this model, it's possible for us to create algorithms to predict disease states. Again, between the rat and the patient with rheumatoid arthritis, we see a similar feature appears to be very important, which is kind of speed of action. The digital biomarkers are more consistent and reliable than some of the subjective scoring techniques. And that it allows us to assess both the functional consequence and the functional response. So is there an improvement in this case? Does the pain reliever have an improvement even if you don't see an actual functional improvement? So with that, I want to finish by talking about 5 key opportunities for digital biomarkers, 5 kind of things that we still need to improve on to improve the use of digital biomarkers in drug development. So the first is around improving preclinical animal models. So digital biomarkers can be used to improve the reproducibility and reliability of our models with the need to understand less about the source of variability. So remember when we talked about in the Cuprizone model, there were many differences across differences across labs in the response, or even when you talk within the same lab as in our case when we were looking at the arthritis and the paw thickness, that there are a lot of factors which can lead to the lack of reproducibility, and that the more data that we get from the animals in the form of these digital biomarkers, the less likely we are going to need to understand all of those sources of variability, because the noise that's associated with those factors, in essence, can be washed away. And we can build ultimately better models that should be more predictive. We're not done with all of the digital biomarkers. So There's still a need for additional sensors, additional biomarkers that can be used across preclinical as well as clinical to track disease. So I talked about a few today. ECGs, for example, and motion are ones that we have in both preclinical and clinical, but there are a number of areas where we're unable to monitor the same things across both. And so there's a need to continue to think about creating those new digital biomarkers or potentially being able to use the same sensors from both human and in animal. And then in addition, clinicians and scientists in the lab, as well as regulatory bodies, really need to work together to validate that those digital biomarkers are valuable biomarkers to be looking at across this whole spectrum of drug discovery and development. So again, there's been a lot of work from the FDA and the NIH about thinking about how to validate those biomarkers across the whole spectrum. Now, there hasn't been a lot of work, or there is a lot of work ongoing in how to validate digital biomarkers in clinic, but we should be thinking more about broadening that back into the preclinical phase as well. To really be able to do this effectively, we need to think about collecting digital biomarkers at scale as well. So in our lab, we've collected over 1 billion hours of digital metric data on many different strains of animals and mice, or rats and mice, many different disease models. What we've learned in the clinic that we need to take back to preclinical. In the clinic, the ability to create these predictive measures has really only come as we're collecting more and more of this data from a wider swath of the population. And so what that really suggests is that the more data we can collect on our animals in the preclinical phase, the more likely we are to be able to create these better models and to be able to translate them or to understand what is translatable or not translatable into the clinic. And then the last is really building platforms or creating platforms that allow us to build better algorithms through AI. So today I talked a bit about some of the digital biomarkers associated with behavioral data in the Cuprizone model. We talked a bit about, or we didn't talk today at all about physiologic data, but we've got data in respiratory and respiratory function as well. I showed you some data from histology. We talked some about manual observations. It's really not until we can collate and collect all of that data into one easy platform that we're going to be able to create some of the best models through AI. And right now, if you look at most labs, manual observations like taking body weights on a scale are collected through Excel spreadsheets. Histology may live in a completely different program. The digital biomarker data, of course, or molecular biomarkers. We really need to be able to integrate all of those together. And really, and I put it in a separate color, is the clinical data as well. So as we start to work more collaboratively between clinicians and researchers at the preclinical stage, how do we take what we're learning in the clinic and actually back translate that into some of the models and potentially collect more digital biomarkers that then again, of course, translates to translatability between preclinical and into the clinic. So with that, I'd like to thank you for your time and attention, and thank you.
Thank you, Dr. Schaevitz, for that informative presentation. I would also like to thank LabRoots for making today's educational webcast possible. Before we go, I want to let everyone know that this webcast will be available for on-demand viewing through May 14th, 2019. As a final reminder, our speaker will follow up with any questions you've submitted via email. That's all for now. Thank you for joining us, and we will see you next time. Bye.